I want to come to the most important point first, and these, of course, are people. I don't go into the individual projects, but I want to highlight names of the active lab and names of people who left the lab in the 5 years. And you have to imagine, every minute I talk, I burn 500K, representing 1 year of ERC funding, so I rather speed up. So the growth. The early retina is small and it still has a complete complement to be functional. So we have a functional retina there, and as it grows, it continues growing, and it doesn't scale like this cut mushroom. No, of course, it grows in its proper form, and that's why I put the cup of cappuccino we're all looking forward to. And you all know that the crema zone here is representing the stem cell niche that goes all around the circumference and adds to the continuous growth of the eye. So the eye grows unidirectionally, grows towards light if you're more a plant person, and that's very easy to memorize. The question is how? What are the components and where is control happening? What is the growth mode? What do the stem cells do that sit in the circumference? How do they contribute to growth? What happens to the two layers, the outer layer, the pigmented epithelium, and the inner layer? How is that balanced? Because they come from the very same stem cell niche. What is happening to stem cells on their way to terminal differentiation? A single stem cell must be amplified to eventually give rise to the full complement that is there. What is happening there? And last but not least, how is shape programmed? Well, we all assume that normally it just continues growing and it keeps its shape. But how is it programmed? This is what we try to address, and I just jumped that. And of course, what we wanted to do is just follow individual cells. This is what we all want, right? We put on the microscope, we look at it, it's beautiful, we get excited, and unfortunately we realize that following the early embryonic development of zebrafish that we've seen in Eileen's movie doesn't give us an answer when it comes to the eye, because here we can image for 24 hours, maybe 48, and if we talk to people in the lab, maybe 72, but not to 2 weeks. And this is what it takes, so we need genetics to really bring it into the game, and the genetics should help to label individual stem cells in that domain, the growth zone. You remember the crema zone? Ideally, you just switch on individual cells, and by switching on, you'll mark the lineage, and you allow to follow all the individual descendants. So switching on color is one thing. Of course, you could imagine not just switching color, but also switch function with color, so that a red cell would behave differently from a green cell. And this is what we've been doing. And we can do that in fish, in genetic systems. You have your wheels of fortune distributed, stochastically activating individual cells all around. And if you're looking at an eye after that treatment, what we'll see is, yes, we get a beautiful rainbow retina, and that rainbow retina, where each color represents an individual clonal string, all descendants from the switched cell, this is what we're getting. So all these cells that originate, that are stem cells, they give rise to these clonal strings that we call arcos, for arc continuous stripes, and all those, interestingly, give rise to all the cell types in the retina. So if we're simply doing a cross-section look through it, all the cells will be represented. So we'll find all 7 cell types, of the new retina in there. The interesting point, and I don't go into it, if we start playing with factors that are crucial for the specification and proliferation here, we can also have stripes, arcos, that are not complete, that have only a subset of that. The point is that those arcos, or those retinal columns, still are functional, because in case of failure, there's always backup other than on the computer. So that works in the retina. So we can really target and tackle all the interactions and find where regulation occurs, and this is what we've been doing. So since they're all multipotent and they're playing in balance, one question was the balance between neuroretina, pigmented epithelium. We can show that it's a pair of genes that, like a yin-yang, is going for either neuroretina or pigmented epithelium. And in the end, we know that these post-embryonic arcos, they are the constituents of the growing eye. And you wonder, strange, why should a fish eye grow like that? This must be a real peculiar system. Of course it is, but if you're looking at vertebrate species all across, except mammals, they all grow that way. So why is that system evolutionary so successful? I think that's a question, and that's a question we try to tackle in combination of the experiments I showed And we wanted to use modeling because doing experiments takes a lot of time. Having a model where you can query kind of the feature space of reality, that should be a way to identify what is more and what is less realistic. And a model runs maybe a couple of hours on the cluster, whereas the experiment takes half a year because you need the ARCOS to grow. So we did that, and by looking at it, and maybe you have spotted already, these ARCOS aren't fine. They're not as nice as the others I showed. They're straight. So you just have to adjust the feature space. This is what we've been doing. And what we find is a very simple thing. We find that if you're looking at the eye like that, so these are kind of staggered cups. The inner cup is the neuroretina. There we have these straight ARCOS. These straight ARCOS are indicative for the shape-giving element. So the inner neuroretina gives shape. The pigmented epithelium is following passively, and the outer layer that eventually also gets pigmented is also following passively. But since we're sharing a stem cell niche here, we see these ARCOS formation in those two things. So I can just wrap up here and say shape is generated inside, the other side is following, and the whole thing still doesn't give an answer on why that is evolutionarily advantageous. So just a hypothesis at the very end. If you grow in these, all you have to trigger, all you have to tweak with is proliferation rate and cell division axis to shift that central part to different positions. Why is that important? That central part is reminiscent of our fovea. So it's the area of highest visual acuity. This is where the fish sees best. And if you're swimming straight, okay, you want it in the middle. But if you're maybe approaching something that is more here, you want it more on the outside. And it just takes a few rounds of simulation and tweaking with proliferation and division axis to get it wherever you want it to. And our speculation hypothesis is that this is the benefit of having a growth in stripes. So this is it, and that's it. Thank you.